lipid, okay, and also the last part which is on nucleic acids, okay. So can you all hear my voice clearly?
Yes.
Yes,
Yes, madam.
Okay, thank you. All right. So basically, for the first part of this lecture, we are going to look at lipids. So lipids, you can there are four types of there are actually four types of lipids, but for your syllabus, we are going to cover three types of lipids. Okay, which are triacylglycerol or triglycerides. Okay, and then the second one is phospholipids, and the third one is steroids. Okay. So what are lipids? So they are a class of large biological molecules that do not form polymers. So lipids are the only macromolecules that are not that we do not consider them as polymers because it is not composed of monomers. Okay, so the lipids are they are made up of mostly carbon, hydrogen, and oxygen. Okay. So the three classes of lipids that we are going to look at are triglycerides, which are fats and oil. Okay, so fats are basically, for example, animal fats. Okay, which will exist in solid state at room temperature. And then the other one, which is oil, usually they are made up of plant oil, okay, which are liquid at room temperature. Same goes to fats from fish, okay, so which are liquid at room temperature. And then the second one is phospholipids, and the third one is steroids. All right. So phospholipid has similar structure to triglyceride, except that on the third carbon of glycerol, it has phosphate group attached to it. Okay. So later we will look at the detailed structure of each of the lipid groups. All right. So the other group of lipid is wax, okay, which we are not going to look at in this chapter. But basically, wax are waterproof substances, right? Um, okay, so what is the property of lipids? So as you know, lipid it will not attract water molecules, so they have no affinity towards water. So therefore, lipids we consider them as hydrophobic. Okay, hydrophobic organic compounds because they consist mostly of hydrocarbons which form non-polar covalent bonds. So as we have mentioned before, hydrocarbons are molecules that are non-polar. Okay, it will isolate themselves. They will isolate themselves from water. It will not attract to water molecule. Okay. And then this lipid will dissolve in organic solvents such as acetone, ether, chloroform, and alcohol. Okay, so the first group of fats here is triglycerides, which are fats and oil. So fats and oil are esters. So in order for you to synthesize fats, okay, they are made up of one molecule of glycerol, which is bonded to three molecule of fatty acids. And the process of you to attach together the glycerol with the three fatty acids is through the reaction of condensation or dehydration reaction. Okay, so and the bond that links together the glycerol with the three fatty acids, we call it as ester linkage. So it means that for one molecule of fats, it will have three ester linkage or ester bond. And then the process of synthesizing the fat, we call it as esterification. Okay, so because it involves alcohol and which attach to acids. Okay, one molecule of glycerol and three fatty acids to form the triglycerides. Okay, so what is actually glycerol? Glycerol is actually a three-carbon alcohol with one hydroxy group attached to each carbon. Okay. So a fatty acids consists of a carboxyl group attached to a long carbon skeleton. So that's why we call it as fatty acids due to the presence of carboxyl group. So I hope that you know the structure of carboxyl. Okay, C double bond C double bond O or H, okay, and then you have the hydrocarbon, okay, attached to the acid. Okay, so this diagram, it shows you the formation of triglycerides. So here is the structure of glycerol. Okay, there. Okay, so this is the glycerol, and then here you have the fatty acids. So in the fatty acid molecule, you will have the carboxyl group. So in order for you to attach the fatty acid with the glycerol, it involves the removal of one water molecule for each fatty acid that you are going to attach to the glycerol. Okay, so it means that in order for you to synthesize the fats, okay, so it requires 3 fatty acid. Okay, so when you attach 3 fatty acid to the one molecule of glycerol, so therefore you will produce 3 molecule of water for every one fatty acid that you attach to the glycerol. Okay, so one water molecule is removed for each fatty acid joined to the glycerol. So this produce triacyl glycerol or triglyceride. Okay, triacyl glycerol, 3 fatty acid with one glycerol. Okay, or triglyceride. Okay, so the bond, you have to know exactly where to label the ester linkage that is formed between the between the fatty acid and the glycerol. So you have to carefully, okay, label the bond. Okay, so the bond will form between the carbon of the carboxyl and the between carbon and oxygen. Okay, of the glycerol here. Okay, so this is the ester linkage. So if in the question, in the exam question, the question asks you to label the ester linkage, you have to label the ester linkage properly. Okay, so if you mislabel, okay, then your answer will be wrong. So if you label something like this, so then it will be wrong. If you label something like this, then your answer will be wrong. Okay. So make sure you take note where to label the ester linkage. Okay, so ester linkage is a bond between a hydroxyl. So this originally was a hydroxy group. Okay, carbon which attached to a hydroxy group. Okay, so and a carboxyl group from the fatty acid. So you have to take note. Okay, how to properly label the ester linkage. All right. So if you look at the structure of the fatty acid here, as you can see, it is made up of carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. So because it is made up of carbon and hydrogen, so they are grouped as hydrocarbon and causes the molecule to be non-polar. Okay, because the bond between the carbon and hydrogen is already stable. Okay, there's no charge, there's no polarity formed, okay, at the fatty acid molecule, so it causes the molecule, this region, to be hydrophobic. Okay, so it has no affinity towards water. Okay, no affinity towards water. Okay, so that's why fat will not dissolve in water. Okay, so this is the basic property of fats. Okay, so non-polar, hydrophobic, no affinity towards water, will not dissolve in water. Okay. So the next one, fats will separate from water. So because a water molecule form hydrogen bonds with each other and will exclude fats. Okay, so as you know, water molecule and with another water molecule, they have cohesive behavior which will attract each other through the formation of hydrogen bonds. Okay, so water will not fats. Okay, fats will not attract water molecule because fat is not polar. Okay, it is not polar. Okay, water will only attract the molecules that are polar. Okay, so fatty acids vary in length according to the number of the carbon along the chain of the fatty acid, and in the number and location of double bonds. Okay, so if the fatty acid contains at least, okay, at least one double bond, we consider the fatty acid to be unsaturated, unsaturated fatty acid. Okay, so if no double bond, if no double bond, okay, so we consider the fatty acid to be saturated fatty acid. So now, what is the difference between fatty acid and if I ask you, okay, what is the difference between unsaturated fatty acid and saturated unsaturated fats? Okay, what is the difference between unsaturated fatty acid and unsaturated fats? What is the difference? The difference is that fat is the whole molecule. Okay, fatty acid is the component within the fat. Okay, so you have to remember fat, okay, consists of glycerol, one molecule of, okay, with three molecule of fatty acid. Okay, so if the question asks you what is the difference between unsaturated fats and unsaturated fatty acid, saturated fat is the whole molecule, unsaturated fatty acid is the component of the fat. Okay, so that you have to take note, make sure you use the right term. Okay, you use the right term. Okay, so here as I've mentioned, okay, so this is the non-polar hydrophobic tail. So they are made up of mostly they are made up of carbon and hydrogen only. Okay, so that's why they are hydrophobic, non-polar. It will not attract water molecule. It causes the fat to be unable to dissolve in water. Okay, so the next one, okay, and the next one that we are going to look at is saturated fatty acids. So saturated fatty acid have the maximum number of hydrogen atoms possibly bonded to the carbon skeleton. Okay, so there will be no double bonds, okay, between the carbon atoms in the hydrocarbon chain, and the molecule is straight molecules. Okay. So example is stearic acid. So as you can see here, so this is an example of saturated fats. Okay, so this is the whole molecule of the saturated fats. Okay, so this is the part where we consider it to be the saturated fatty acid. Okay, so saturated fatty acid. So here, example of saturated fats given in the example is butter. Okay, so which is animal fats. Okay, so fats made up of saturated fatty acid are called saturated fats and are solid at room temperature, such as butter and lard. Okay, butter is the fat from milk. Okay, and then lard is the fat from the sauce that we cannot eat. Okay, daripada babi lah. Okay, so we cannot eat. So you have to really make sure that when you eat pastry product, for example, it does not contain any lard. Okay. Next, okay, so because the hydrocarbon of fatty acids lack double bond and allows the fat molecule to pack together closely, so most animal fats are saturated. So why is it that saturated fats are solid at room temperature? This is due to because there is no double bond present in the fatty acid chain of the fat molecule. So it allows the fats to be packed closely together. So when the molecule of fats, okay, packed closely together, it causes the fat to exist at solid state, okay, at room temperature. So for example here, so this is one fat molecule. So this is another fat molecule. So this is another fat molecule. So this is another one, another one, another one. Okay. So this causes the fat to be solid. Okay, solid at room temperature. Okay. So why? Because due to the fatty acid molecule here, they exist as straight chain. Okay, there is no bent structure present. Okay, so that's why saturated fat will be solid at room temperature because the molecule of fat can pack closely together because there is no double bond. If for example you have double bond here, okay, double bond here between the carbon, and then obviously when there's double bond between the carbon, it forms unsaturated fatty acid, and this will cause, okay, the molecule, okay, the fatty acid here to be bent, the molecule to be bent. So if the molecule is bent, okay, so this will prevent the fat from packing close together. Okay, so this is the explanation for the unsaturated fatty acids. Okay, so unsaturated fatty acids will have one or more double bonds. Okay, they have at least one double bond. Okay, so where you have the double bond, okay, it causes the fatty acid molecule to be bent. Okay, so this is carbon, for example, this is another carbon along the chain of the fatty acid. So the carbon are bonded by double bond here. Okay, so you have hydrogen here, hydrogen here. Okay, so carbon, hydrogen, hydrogen, hydrogen. Okay, so where you have the double bond between the carbon, so the each of the carbon will have less hydrogen atoms. Okay, so that's why we call it as unsaturated. So the fatty acid molecule does not have the maximum number of hydrogen. Okay, so that's why we call it as unsaturated, right? As compared to saturated, they have the maximum number of hydrogen atoms. So that's why we call it as saturated fatty acid. Okay. So here it says where you have the double bond, it causes the molecule to be bent. Okay, the bent structure we call it as kink. Okay, kink. So we'll have kink in its hydrocarbon chain wherever a cis double bond occur. So what is cis double bond? Kita belajar kan kat functional group. Cis double bond is where you have the functional group or group of atoms that are located on the same side. Okay, so if trans what the functional group kan yang X tu, so the functional group are located, okay, at the opposite end here. Okay, so like this one is trans, this one is cis. Okay, so for unsaturated fat, they usually will have cis double bond. So example is oleic acid and linoleic acid. Okay, so unsaturated fatty acid is the component of unsaturated fats. So here is an example of unsaturated fat molecule. So it has three fatty acids. Okay, it has three fatty acids. So the three fatty acid, one of the fatty acid will have one double bond. Okay, so although it has only one double bond, so we consider the fat to be unsaturated fats. Okay, so due to the presence of the double bond, it causes the fatty acid molecule to be bent. Okay, and therefore what happened is that it says here fats made up of unsaturated fatty acid are called unsaturated fats or oil and are liquid at room temperature. Why is it liquid? Because the molecule is bent and causes bent structure, kink. Okay. So when you have kink, okay, so this will prevent the molecule from packing together closely to solidify. That's why they exist in liquid. Okay, liquid form. Okay, liquid form. So due to the presence of the double bond. Yes, you have kink. Okay, so where you have kink, so they cannot pack closely together. Okay, ada double bond kan. So this will prevent the molecule from packing closely together. So they exist as liquid at room temperature. So fats from plant, okay, or fish are usually unsaturated. Okay, so it says here a diet rich in saturated fats may contribute to cardiovascular disease through plaque deposition within the blood vessels, which will block the flow of blood. Okay, so why? Because the fats will solidify in your blood vessels and causes the formation of plaque. Okay. So the next one is dehydrogenation. Okay, dehydrogenation. So a process of converting unsaturated fats. Okay, usually plant oil ataupun vegetable oil. Okay, vegetable oil to saturated fats by adding hydrogens. Okay, so for example, what you want to make margarine. Okay, margarine. So margarine is the solid form of vegetable oil. So normally vegetable oil will exist as liquid. Tapi bila nak sapu kat awak punya roti macam tu, takkan nak sapu minyak yang cair tu kan. So you want to use margarine. So margarine is the product from the hydrogenation of vegetable oil. Okay, so hydrogenating vegetable oil will create saturated fats and unsaturated fats with trans double bond. Okay, trans double bond. Okay, trans double bond. So it forms something like this. Okay, so so this is the normal cis double bond. Okay, so where you have the functional group. Okay, which is on the same side. Now they are on the opposite side. Okay, so when you produce a double bond here along the fatty acid chain, it causes originally, okay, you have a band structure. Okay, band structure, band. Inilah macam tu kan, macam tu band. Okay, so by through hydrogenation, it causes cis double bond to transform into trans double bond and causes the fatty acid to be transformed into a straight structure. Okay, so this, okay, will contribute to cardiovascular disease also. Okay, so these trans fats may contribute more than saturated fats to cardiovascular disease. So example of food that contains trans fats are baked foods, okay, and also processed food. So make sure to avoid food that contains trans fat. So usually if you look at your food, the label at the back, it will label whether your food contains trans fat or not. So if it does, so try to avoid. Okay. So the next one is phospholipids. So this is the structure of a phospholipid. So as you can see, it has the hydrophilic head region, the hydrophobic tail region. So the hydrophobic tail region is made up of fatty acid, okay? And then the hydrophilic head is made up of glycerol and also phosphate. Phosphate group. Okay, and also a choline group. Okay, so as we have learned before in the subtopic of functional group, you know that phosphate functional group, it is polar. Okay, why? Because phosphate will have a negative charge, okay, on its structure. So due to the presence of the negative charge at the phosphate group, which is located at the hydrophilic region, it causes this region to have affinity towards water. Okay, this one, because it is made up of fatty acid, so it is hydrophobic, does not attract water. Okay, so phospholipid molecule has two fatty acid attached to glycerol, which forms the hydrophobic tail. Okay, hydrophobic tail. And the third hydroxy group of the glycerol is joined to a phosphate, and its attachment is referring to the presence of choline group, which has a positive charge. Okay, which has a negative electrical charge. This is referring to the phosphate, okay, which forms hydrophilic heads. Okay, so this is a region that have affinity towards water. Hydrophobic tail will exclude from water. It does not attract water because it is non-polar. Okay. So this is the structure of a phospholipid. So as you can see, it has a glycerol, okay, glycerol with 3 carbons, and 2 of the carbon is bonded to each is bonded to one molecule of fatty acids. So for example, here you have a double bond exists, so it causes the molecule to be bent or kink. Okay, bent, right? So in this case lah. And then in chapter 3, okay, in chapter 3, we will look at what is the significance of having double bond, okay, in the fatty acid chain of the of the hydrophobic tail of the phospholipid. The other function there, it has its own significant role, okay, in your plasma membrane. So that one we are going to look at in chapter 3. Okay, so the third carbon is bonded to phosphate. So phosphate has negative charge here, okay, and then a choline group, okay, which has positive charge here, okay. So this causes this portion to be hydrophilic, okay, it will attract water, right? So when phospholipids are attached to water, so they will self-assemble into a bilayer with the hydrophobic tail pointing towards the interior. Okay, so hydrophobic tail will attract each other, okay, and they will be excluded from the aqueous solution, okay, from the surrounding aqueous solution. So the structure of phospholipid results in a bilayer arrangement found in cell membrane. Okay. So phospholipids are the major component of all cell membrane. So this is the phospholipid bilayer of membrane. Okay, so membrane. So either it is a plasma membrane of cells or membranes of organelles. So membrane of organelles are also made up of phospholipid bilayer. Okay. So as you can see, the hydrophilic heads are facing the aqueous solution. Okay, aqueous solution. Okay, so if this is a plasma membrane, okay, so outside can be the extracellular fluid, inside can be the cytoplasmic region. Okay, cytoplasmic region. So both region are aqueous. Okay, so they have water. Okay. So the hydrophilic heads are on the outside of the bilayer in contact with the aqueous solution. Okay, inside and outside of the cell. Inside is cytoplasmic region, outside is the extracellular fluid side. Okay, so and then the hydrophobic tails will attract to each other. Okay, they will point towards the interior of the bilayer away from the water. So this is hydrophobic interaction. Okay. So hydrophobic interaction, if it exists alone, so it is a weak bond. But if you have so many of the hydrophobic interaction, so it can form a strong interaction. Okay. Okay, so the third class of lipid is steroids. So steroids are lipid characterized by a carbon skeleton consisting of 4 fused ring. Okay, 4 fused ring. So that is steroids. Okay, so it has 4 fused ring. 1, 2, 3, 4. So this one you have to remember. Yes, dia adalah antara soalan yang suka keluar lah. So you have to know the structure of a steroid. Okay. So although it does not contain fatty acid as triglyceride do, and phospholipid also have fatty acid. So why is it that steroid we consider them to be lipid? Okay. So this is because it is insoluble in water. Okay. It is non-polar. Okay. It is non-polar. It can only dissolve in non-polar solvent. Okay. So as you can see, why is it that steroid, it is insoluble in water? So because it is made up mostly of carbon and hydrogen. They are hydrocarbons. Okay, hydrocarbons. So hydrocarbon, okay, where you have the bond between the carbon and hydrogen are very stable, so it causes the molecule to be non-polar. Okay, non-polar. So non-polar, it will not attract water. Okay, so that's why steroid is considered as lipid. Okay, it will not dissolve in water. So so there are many types of steroids. Okay, so how to differentiate between one steroid to another steroid to another steroid? By looking at the side chain. Okay, so so the most common steroid this is the structure of cholesterol. Okay, cholesterol. So cholesterol is a type of steroid that are most commonly found in your cells. Okay, so because you can find cholesterol embedded in the plasma membrane. Cholesterol also is the initial molecule or precursor molecule to synthesize your sex hormone, okay, hormones. So testosterone, for example, estrogen, progesterone, sex hormone, of which it is initially synthesized from cholesterol. Okay, so this is cholesterol. So cholesterol is an important steroid. It is a component of animal cell membrane. You can only find cholesterol in animal cell, not plant cell, not plant cell. Okay, so cholesterol has its own significance why it has to be embedded in the plasma membrane. So yang itu kita akan cover masa chapter 3. Okay, what is the role of cholesterol in the plasma membrane? Okay, so it is the precursor, okay, precursor from which other steroids are synthesized. So many hormones, including vertebrates, sex hormones, are steroid produced from cholesterol. Okay, sex hormone, estrogen, progesterone, testosterone. Those are easy example of the sex hormone. Okay, so this is the side chain of cholesterol. So if you want to find what is the how does the side chain of testosterone looks like, so you have to search yourself lah. Okay, so just look at the difference what between the structure of side chain of other steroids. Okay. Okay, so the next one, the next subtopic is nucleic acids. Okay, so before we proceed with the nucleic acid, do you have any question regarding on the lipid topics? Ada soalan? Ada ke tidak? Ke tidur depan depan komputer ni?
Tak ada, madam. No,
Ada.
madam.
Okay, so faham ada.
Madam, yang for few string tu yang mana?
1, 2, 3.
Yang tu.
Ya?
Sebab saya dengan member saya confuse.
Ni, for few string. Okay.
Few strings tu composed of what, madam?
Carbon, hydrogen, carbon, each one is kan?
Okay, thank you.
Okay. So that's why there are hydrocarbons. Okay, hydrocarbon tadi, non-polar. So if there's no more questions, so we are going to proceed with the next subtopic, the last one, which is the nucleic acids. Okay, so it says here, okay, so the amino acid sequence of a polypeptide is programmed by a unit inheritance called gene. Itu ayat pertama dia. Faham tak? Faham ke tidak? Apa ayat ni nak cakap? The amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. Ayat introduction dia pun dah susah dah. Tapi faham ke tak? Apa yang dia nak cakap kat sini?
Tak faham, madam. Faham, madam.
Faham?
Faham,
madam.
Oh.
Okay, bagus lah, faham. So saya tanya explain Okay. So saya explain lah. So what does the sentence means? Okay, the amino acid sequence of a polypeptide is programmed, okay, by a unit of inheritance called a gene. So your cells, okay, your cells will require so many different types of proteins. Okay, for example, in your cell you have different enzymes. Okay, your cell will produce different types of hormones that are made up of proteins. Your cell also requires structural proteins, okay, such as collagen, such as microtubules. So those are proteins, okay. In order to synthesize those proteins, it requires information that are stored in the nucleus, okay, which are stored in the gene in the nucleus, okay. Gene in the DNA which is inside your nucleus. Okay. So here, tu saya dah terang dah di kat awak. Dah terang dah kan? Ke belum?
Belum.
Belum. Okay. So this is cell. Okay. Cell. So inside your cell, I've just left the meeting. Sekejap. Saya nak kena rejoin balik. Okay. So this is a cell. So inside cell, you have nucleus. Okay. So this is a nucleus. So the nucleus, okay, inside the nucleus you have a large molecule which we call it as DNA. Okay. So the DNA, as you know, it stores genetic information. So this genetic information, okay, each of the information is stored in segments, okay, within the DNA. We call it as genes, okay. Genes are segments of DNA that code for the synthesis of certain protein, okay. So for example, here, so this is one region that that we label it as gene 1, okay. So this is another region, we label it as gene 2. Another region, gene 3 and gene 4 and so on, okay. So each of the gene will code for the synthesis of certain or specific protein. Okay, so your cell will produce so many different types of proteins such as enzyme. You have so many different enzyme, hormones. Okay, hormone, you have so many different hormones. You have structural protein. Proteins, okay, structural proteins such as microtubules, okay, microfilaments, collagen, okay, actin, myosin. So those are different type of proteins that your cell will produce, okay. So how to produce these hormones, okay? So they are coded in the gene that are found in your DNA inside the nucleus, okay. So but the synthesis of protein, it occurs where? In the cytoplasm, okay. But the instruction is within the nucleus. So how can protein synthesis occur in the cytoplasm? So you have to copy the information, okay. Copy the information. In order for you to copy the information, you have to copy the information into a smaller structure. Macam contohnya awak punya komputer has all your data, has all your information, tapi awak nak extract out certain information je keluar daripada awak punya komputer tu. Awak guna apa? Contohnya awak guna pen drive kan. So smaller version, okay. So kat sini, okay, let's say lah example, at one moment, okay, the cell want to produce enzyme A. Okay, enzyme A. Enzyme A is coded within the gene of gene 2 here. Okay, enzyme A. Kalau gene 4, let's say lah other enzyme B for example. Okay, so the information is within the gene. So you copy the information in the form of mRNA. Okay, in the form of mRNA. So mRNA is a smaller molecule, okay, that has the same information which is it code for the synthesis of enzyme A. So as you know, DNA is double-stranded. mRNA is a smaller molecule which is single-stranded. So because the molecule is much smaller, Okay, so you have to know that the nucleus, okay, the structure of the nucleus, it has double membrane. Double membrane, sorry lah, tak berapa cantik. So within the membrane of the nucleus, they have pores. Nuclear pore, we call it. Okay, nuclear pore. They have pores. Nuclear pore, so many all the way through lah. Okay, nuclear pore. So nuclear pore. So by having nuclear pore, it allows the mRNA to exit the nucleus and enter into the cytoplasm. What can you find in the cytoplasm? You have another organelle, which is ribosome. So this is ribosome. Okay. So what is a ribosome? Ribosome is an organelle which is involved in protein synthesis. Organelle that involve in protein synthesis. Okay, so a ribosome is made up of large subunit and small subunit. So these large and small subunit are large and small subunit, large yang atas, small kat bawah. So it is made up of protein together with rRNA, another type of RNA. rRNA stands for ribosomal RNA. Okay, mRNA stands for messenger RNA. Messenger RNA, why? Because it copies information from the DNA. Okay. So next, okay, as mRNA migrates, okay, from the nucleus into the cytoplasm, it will find a ribosome and it will enter in between the region of large and small. Okay. So what else can you find in the cytoplasm? Okay, in the cytoplasm, you have abundance of amino acid. Okay, you have abundance of amino acids. You have the different, okay, the different types of amino acids. Okay, inside your cytoplasm. Okay. So another type of RNA that can be found in the cell. Okay, you have tRNA. Okay, what does T stands for? T stands for transfer RNA. So it transfer what? It transfer amino acids. Okay, so ribosome here, okay, it will code the code on the mRNA. So as you know, okay, DNA and RNA are made up of monomers. We call it as nucleotides. Okay, DNA and RNA are made up of monomers called nucleotides. Okay, there are different nucleotides based on the bases that are found on the nucleotides. So kita ada apa? Kita ada A, T, C, G kan dalam DNA. Okay, and for RNA we have A, U, C, G. Okay, so the sequence of the nucleotides along the DNA will be different. Okay, will be different. Dia boleh jadi A, U, G, G, C, C, A, T, G, whatever lah along the chain kan. So the information will be copied in the mRNA. Let's say for example, A akan match dengan apa? A dengan T kan, tapi sebab dia mRNA dia jadi U. U dengan A, G dengan C, C, G. Okay, so this is for example the sequence of nucleotide on the mRNA. Okay, so this sequence will be read by the ribosome. So the sequence, okay, we call it it codes for specific amino acids. Okay, every three nucleotides code for one amino acid. It codes for one amino acid. So the ribosome will read the code on the mRNA, and what happens next is that the tRNA will bring specific amino acid to the ribosome. Okay, to be that is being read. Okay, so in the process of synthesizing the protein, okay, so protein amino acid are added to the chain by tRNA, bringing that specific amino acid to the ribosome. Okay, so this is the polypeptide chain. Okay, the tRNA will bring specific amino acid according to the code on the mRNA. So this is the polypeptide chain. Okay, composed of amino acids. Okay, so polypeptide chain yang ni lah nanti dia akan hasilkan the specific protein that your cell requires. So in this case, enzyme A. Okay, so this is the structure, the initial structure of enzyme A. Okay, so that's why itu adalah penerangan untuk yang statement ni, satu ayat je. But the whole process is really long. Okay. Tapi the detailed process actually we are going to learn in chapter 1 of next semester, okay, which is genetic. So that is a whole different story lah. But saya explain tu sebab nanti kita akan tengok kita ada mRNA, tRNA, rRNA. So roughly you know the function of each different RNA. Okay. So the amino acid sequence of a polypeptide is programmed. It means that it is coded, okay, by a unit of inheritance called gene, okay, which is a segment along the DNA that code for specific protein. Okay. So gene consists of DNA, okay, which is a nucleic acid made of monomers called nucleotides. Okay, so this is the structure of a nucleotides. So you have to know one nucleotide is composed of a pentose sugar, a 5-carbon sugar, bonded to a phosphate, and then it is bonded to a base. So base, okay, we have for DNA, ATCG. For RNA, kita ada AUCG. Okay, so there is no thymine in the DNA. Okay, so for the 5-carbon sugar, okay, for DNA, it is composed of deoxyribose. Ni dalam satu ayat dalam satu perkataan deoxyribose. This one is ribose. Okay, so ribose. So this is basically the structure of a ribose. Okay, because at carbon number 2 you have hydroxyl, but for deoxyribose it only has hydrogen. Okay, so how do you number the carbon? So this is carbon number 1, carbon number 2, number 3, number 4, and number 5. So it is a 5-carbon sugar. So the 5th carbon is bonded to a phosphate group. Okay, so as you can see, phosphate, it has a negative charge. So the formula apa? PO4 2 negative. Nampak? PO4 2 negative. So why does it has a negative sign? Because originally, okay, OO, it has hydroxyl here. Okay, so when your DNA exists in your cells, so it is in aqueous solution. Okay, so the molecule will be ionized. Okay, when the molecule is ionized, it releases the hydrogen ion into the solution. Okay, so by releasing the hydrogen ion, so it causes the DNA to have an acidic property. Okay, so that's why we call it as deoxyribonucleic acid. Okay, due to this the presence of phosphate that releases hydrogen ion when it ionizes in your cell. Okay, dalam bentuk aqueous solution, so dia akan release H+. Okay, so that's why it is called as deoxyribonucleic acids. Okay, or ribonucleic acids for RNA. Okay. So a base yang tadi lah ATCG or AUGC. So there are 2 types of nucleic acid, DNA, deoxyribonucleic acid, and ribonucleic acids, RNA. Okay, so DNA provides directions, okay, for its own replication. So during cell division, such as for example mitosis or meiosis, okay, so DNA will replicate themselves. Okay, so that the same DNA will be distributed into daughter cells. Okay, the same genetic information, okay, is distributed to the daughter cells, especially if it involves mitosis. Okay, And then DNA directs synthesis of mRNA. Tadilah saya dah tunjukkan mRNA. From DNA, it synthesizes mRNA. And through mRNA, control protein synthesis because mRNA copies the information from the DNA. Okay, so protein synthesis occur in the ribosome, in the cytoplasm. Okay, so for RNA, you have three types: messenger RNA, transfer RNA, tRNA, rRNA, ribosomal RNA. Okay, so dalam gambar tadi, as I've drawn earlier, okay, so you see that tRNA and RNA are found in the cytoplasm, but actually, okay, all the three RNAs are produced in the nucleus. Okay, they are produced in the nucleus, okay, according to the instruction from the DNA. Alright. Okay, so in the DNA, the sugar is deoxyribose. DNA, the sugar is deoxyribose. So this is the structure of deoxyribose. Okay, so this is carbon number 1, carbon number 2, carbon number 3, number 4, and number 5. Okay, so kat sini ada oxygen. Alright, so for carbon number 2, you have hydrogen atom. Okay, so this one is for deoxyribose in DNA. But as for RNA, the ribose sugar, at carbon number 2, it has hydroxyl. Okay, ribose. So the other structure, they are the same, okay, except for at carbon number 2, right? So deoxyribose, because why? Because it is lacking an oxygen. Dia ada hydrogen atom sahaja, right? So this one at carbon number 2, it has OH, hydroxyl. It has extra oxygen, okay? So nucleoside components. So this is nucleotide lah. This is nucleotide. For nucleoside, this one, sugar and base only, nucleoside. Okay, so that's why kat sini di label as nucleoside component, sugar and base. Okay, bila tambah dengan phosphate, baru kita panggil dia sebagai nucleotide. Right? Okay, the structure of inilah, okay. So nucleic acids are polymer called polynucleotides. You add nucleotides together along the chain. So you have so many nucleotides. So they are polymers. So they are polymers of polynucleotides or nucleic acids. Okay. So each nucleotide is made up of monomers, which is nucleotides. So each nucleotide consists of three components: a nitrogenous base, pentose sugar, and a phosphate group. Okay. So nucleoside, okay, sugar and base only. The portion of a nucleotide without the phosphate, okay, that is nucleoside. Nucleoside, sugar and base only. When you add phosphate, then only nucleotide, okay. Okay, so this diagram, it shows you a polynucleotide chain, okay. So in this chain, it connects between 1, 2, 3, 4 nucleotides, okay. And as you look at this diagram, okay, you will notice it is labeled as 5' N and 3' Ns. So what does it mean? So it means it shows you the directionality of the polynucleotide chain. Okay, polynucleotide chain, especially DNA, it has a directionality. Dia akan ada direction kat situ. It shows you the direction of the DNA, okay, to be read along the ribosome, for example. Okay, so this is 5' N and 3' N. How do you know which end is which? Okay, by looking at the 5 carbon sugar. So you have to know again how to number the 5 carbon sugar. So this is carbon number 1, carbon number 2, number 3, number 4, number 5. Carbon number 5 is always bonded to a phosphate group, okay, of its own within its own nucleotide, okay, structure. Okay, so this is one nucleotide. This is the second nucleotide. Okay, this is the third nucleotide, the fourth nucleotide. Okay, so as you notice, okay, the last nucleotides, the 3' N is free. Okay, carbon number 3 is free. So carbon number 3, it has hydroxyl group bonded to it. Nampak kat sini kan? So this is carbon number 3. Number 1, number 2, number 3. Carbon number 3 will have hydroxyl group. Okay? Will have hydroxyl group. So what is the significance of labeling the 3' end? So if, for example, during DNA replication, okay, in order for you to synthesize DNA, the new nucleotides, okay, when you want to add new nucleotides, if you want to add new nucleotides, you will always have to add at the 3' end. Okay, so the reaction involves carbon number 3. You cannot add the new nucleotide at the 5' end. So because the 5' end is already bonded to its own phosphate. So the carbon number 5 is not free for a reaction to occur. Okay. So in this case, it involves the reaction between carbon number 3 and the phosphate of the next nucleotide to be added. Okay. So the next nucleotide to be added, for example, here, okay, phosphate, this is the pentose sugar, and this is the base. Okay. So phosphate, as you can see, phosphate here, okay, it will have yang OH kan, asalnya OH kat sini kan. Okay, OH. So in the reaction where you want to attach the two nucleotides together, again it undergoes the same reaction as you want to attach monomers of polypeptide or polysaccharides. The same reaction it undergoes, which is condensation or dehydration. So the process release water molecule. So in this case, okay, in the formation of the bond between the two nucleotides here, it involves the release of water molecule. So it means OH will be released, H from O will be released to form the water, and you will form the bond between the third carbon of the last nucleotide and the phosphate of the next nucleotide. Okay, so this is, we call it as phosphodiester bond. Okay, this is phosphodiester bond. Okay, the bond between nucleotides in the DNA is, we call it as phosphodiester bond. Okay, phosphodiester bond. Right. So before we proceed to the formation of the phosphodiester bond, you have to know, okay, for nitrogenous base, okay, which is bonded to the 5 carbon sugar, you have 5 different bases: cytosine, thymine, uracil, adenine, and guanine. Okay, so the structure for cytosine, thymine, uracil, it is a single six-membered ring. 1, 2, 3, 4, 5, 6. 6-membered ring, a single 6-membered ring. But cytosine, thymine, and uracil, they are pyrimidids. Okay, so for purines, ada adenine and guanine saja. Adenine and guanine. So it is a 6-membered ring fused to a 5-membered ring. 6-membered ring fused to a 5-membered ring. So this one you have to remember: pyrimidine, cytosine, thymine, uracil, purine, adenine, and guanine. Okay. So the DNA, as you know, it is double-stranded kan, helical structure. Okay. So in order for the helical structure to hold its structure as helical, dia tak terputus, so you have to have bonds, okay, that hold the two strands together. So the bonds are hydrogen bonds. Okay. The bonds are formed between the bases. Okay. The bases. Let's say on this strand, you have adenine, and the other one you have it has to match thymine on the other strand. This one you have guanine, on the other strand it has to have cytosine. Okay, dia tak boleh adenine and guanine, tak boleh So adenine has to pair with thymine, guanine has to pair with cytosine, and the hydrogen bonds that are formed between thymine and adenine, you have 2 hydrogen bonds. Between guanine and cytosine, you have 3 hydrogen bonds. This one you have to remember. Okay, so So in order to hold the two strands of the double helix together, it is held by hydrogen bonds between the bases. Okay, between adenine and thymine, guanine and cytosine. So this is what we call it as complementary base pairing along the DNA. Okay. So the next one, okay, the formation of the of the phosphodiester bond. Okay, this is a phosphodiester bond. So phosphodiester bond is a type of covalent bond. Okay, because it is the bond that exists within DNA molecule ataupun RNA molecule. Okay, so you have to know that covalent bond exists within atoms within a molecule. Okay, DNA is a molecule, RNA is also a molecule. Okay, but DNA is a large molecule. Okay, so a type of covalent bond between adjacent nucleotides, the first nucleotides and the second nucleotide here in this case. Okay, so formed between the hydroxyl group of carbon number 3, carbon number 1, number 2, number 3, number 4, number 5. So carbon number 3 originally it has hydroxyl. Okay, it has hydroxyl. Originally it has hydroxyl. So of one nucleotide and phosphate on the fifth carbon of the next nucleotide. So this is the next nucleotide. 1, 2, 3, 4, 5. So carbon number 5 of the next nucleotide has its own phosphate. Okay. So the reaction involves what? Involves the removal of water molecule. So it is a type of condensation reaction or dehydration reaction. So the chain of nucleotide has a 5 to 3 prime orientation. Okay, this is what we call it as 5 to 3 prime orientation. Okay, 5, 5 prime N and 3 prime N. So we call it as 5 to 3 prime N. Okay, 5 to 3 prime direction. Okay, so whenever, for example, you want to synthesize protein, okay, so the mRNA, okay, that is read in the ribosome, it is always read in the 5 to 3 prime direction. Whenever you want to synthesize DNA or RNA, it is always in the 5 to 3 prime direction. You always add new nucleotide at the 3 prime end. Okay, the mRNA is always read in the 5 to 3 prime direction. So that's why we call it as 5 to 3 prime orientation or direction. Okay, so the backbone is negatively charged due to the negative charge on each phosphate. Okay, so this is the backbone of the DNA. Backbone. So the blue color here is the backbone. So it is a repetitive unit of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and sugar. So that is the backbone of DNA. It consists of phosphate and sugar. Okay. And protruding from the backbone, you have the base. Okay, protruding from the backbone, you have the base. Okay, so the DNA double helix. A DNA molecule has two polynucleotide strands spiraling or coiling together around an imaginary axis, forming a double helix structure. Okay, so imaginary axis. Okay. So they will coil around one another. So in the DNA double helix, the two backbone run in the opposite direction, okay, which is 5 to 3 prime direction. So this is what we call it as anti-parallel. Okay, so for example, kat sini kan, so this is the first DNA strand. So this is the second DNA strand of a DNA molecule. Okay, the first strand and the second strand. So if the first strand it reads 5 prime, so the complementary end is 3. Okay, 5 has to match with 3, and this end is 3, and this end is 5. So the directionality, okay, the top strand will run in this direction, the second strand will run in this direction. Okay, 5 to 3, 5 to 3. Okay, so this is we call it as anti-parallel. Okay, anti-parallel. Okay, they run in the opposite direction. The two strands of DNA runs in the opposite direction. Okay, so one DNA molecule includes many genes. Like we have looked before, you have so many different genes that codes for so many different proteins. Okay, the protein can end up being different types of protein, enzyme, hormones, or whatever lah that your cell requires. So the nitrogenous bases in the DNA pair up and form hydrogen bonds. So if this strand here, you have adenine, so it has to pair with thymine. If this has guanine, it has to pair with cytosine. Okay, sama lah along the chain. Okay, C, for example, G, T. So yang ini kena G, C, A. Okay, so that is what we call it as anti-parallel. Okay, anti-parallel opposite orientation, and the pairing between the bases we call it as complementary pairing. Okay, so so you have bonds between the bases, which is hydrogen bonds that hold the two DNA strands together, right? So adenine and thymine, 2 hydrogen bonds, guanine and cytosine, 3 hydrogen bonds. Okay, like this one. Okay, pernah keluar dalam exam? Disuruh awak label hydrogen bonds between the bases. So you have to know adenine and thymine, 2 hydrogen bonds. So again, every time if you want to draw a hydrogen bonds, you must use dotted lines. Okay, so adenine, thymine, 2 hydrogen bond, 1, 2. Okay, cytosine and guanine, 3 hydrogen bond, 1, 2, and 3. Okay. Okay, so basically, okay, so this is DNA. Okay, so DNA, adenine paired with thymine, cytosine paired with guanine. You have hydrogen bonds that hold the two DNA strands together. Okay. So as you know, hydrogen bonds, they are weak bond, but if it exists in so many amount, okay, so it gives a strong structure to the DNA molecule. Okay, so nanti masa chapter 1 semester depan kan, kita akan belajar satu proses, kita namakan dia sebagai PCR, okay, polymerase chain reaction. So this is to duplicate DNA in large amount. So in the process of PCR reaction, you will apply very high temperature. Why do you apply a very high temperature? This is to break the bond between the two DNA strands, because they are hydrogen bonds. Okay, so you can break hydrogen bonds using heat. Okay, using heat, high temperature. Okay, you have to heat the DNA at high temperature in order to break the hydrogen bonds between the two DNA strands, so that you can produce separate DNA strand as template. Okay, as template to synthesize new DNA. Okay, so yang this one is tRNA. So tRNA ataupun RNA, as you know, it is a smaller molecule and they are single strand. Kalau DNA, double strand. Okay, so tapi awak tengok kat sini, madam, tapi ni macam double strand je kan, nampak macam double strand, dia ada pairing, but it is actually a single strand. The only thing is that for RNA, they akan fold, okay, into specific shape. Okay, wherever you have folded area here, okay, you have pairing between the bases. Okay, so tRNA is actually a single strand, but they are folded into a specific structure to form the structure of tRNA, so that here you have the region where the amino acid will bind. Okay, so here you have anticodon region. Anticodon region tu kita akan belajar nanti semester depan lah. Okay, so tRNA, this is the structure of tRNA. It has two regions. The region where amino acid will bind, and another region we call it as anticodon. Okay. Okay, so this table shows you the difference between DNA and RNA. So soalan ni adalah antara soalan yang sukalah keluarkan dalam soalan structure. Okay, so you have to know lah the difference between DNA and RNA. So DNA consists of a single strand only, and they are much shorter compared to DNA. Okay, because RNA is synthesized from information in the DNA. Okay, it copies information from the DNA. So DNA is a double strand Okay, they are arranged in anti-parallel and they are coiled together. Okay, kalau double, whenever you write, bila soalan keluar untuk awak differentiate, make sure awak punya perbezaan tu tally. Maksudnya kalau sini awak bezakan single strand, ini mestilah double strand. Faham? Baru boleh dapat 1 mark. Okay, tak boleh lah single strand lepas tu nak comparekan dia dengan contohnya larger molecule. So dia tak match lah, faham tak? So walaupun facts awak betul, kan? Facts dia tak salah, tapi you are comparing two different things, so you will not get mark for that. Okay, so make sure whenever you answer this type of question, you answer in the table form. Dah keluar, okay tak apa, awak tengok awak punya nota je. Awak tengok nota awak je. So whenever you answer this type of question, make sure that you answer in a table form and you compare apple to apple lah. Tak boleh apple to pear kan, so dia jadi salah lah. Okay, so the next you just look at your notes. Okay, the next difference is that for RNA it has a pentose sugar which is ribose. DNA it has deoxyribose. So that is the second difference that you can write. And then for RNA, the bases are uracil, adenine, guanine, and cytosine. Okay, kalau DNA you have thymine, adenine, guanine, and cytosine. So tak boleh ada uracil dalam DNA, tak boleh ada thymine dalam RNA. Okay, so number 4, produced in the nucleus but found anywhere within the cell. So I've mentioned that all the 3 RNA, mRNA, tRNA, and rRNA are produced in the nucleus, but because they are involved in protein synthesis, and protein synthesis occurs in the cytoplasm, so they are transported. So these three RNAs will be transported in the cytoplasm. So that's why you can find RNA throughout the cell, in the nucleus and also in the cytoplasm. But as for DNA, it is only found in the nucleus. Okay, found almost entirely in the chromosome within the nucleus, and there are 2 organelles that have their own DNA, which is mitochondria and chloroplast. Mitochondria and chloroplast will have their own DNA. Why? Because mitochondria and chloroplast will involve in major process within cell, which is mitochondria. It involves in what process? Cellular respiration. Photosynthesis, chloroplast involves in photosynthesis. These 2 major reactions within cells involves a series of chemical reactions which are catalyzed by many different enzymes. Okay, and in order to synthesize this enzyme, it has to come from instruction of DNA within the organelle. Okay, so mitochondria and chloroplast will have its own DNA because they have to synthesize many enzymes that involve in the chemical reaction of cellular respiration and photosynthesis respectively lah. Okay, and because mitochondria and chloroplast have their own DNA, so the mitochondria and chloroplast will replicate themselves without the instruction from the nucleus. Maksudnya mitochondria ni dia boleh membahagi dengan sendirinya. Okay, sebab dia tak bergantung pada instruction daripada nukleus. Okay, so DNA you have three types: mRNA, tRNA, and ribosomal RNA. For DNA you only have 1 type. Okay, so RNA has smaller nuclear mass. DNA has larger nuclear mass. Okay, because DNA is a much larger molecule within the nucleus, and then RNA does not act as a gene. It is important in the production of a protein. So only DNA will have genes. Okay, DNA will have genes that code for the synthesis of certain protein, but the information from the gene is copied into the RNA. Okay, so the function of RNA is to copy the information and involves directly in protein synthesis in the nucleus. Okay, kalau DNA is just to code the information. Okay, kalau RNA, it involves directly in the protein synthesis. So the last slide is to summarize the whole of chapter, the whole of macromolecule lah. Okay, so you can do your own mind map. Okay, so kalau rasa ni dia just rabut, so just ignore, you do your own mind map. Okay, according to your own understanding lah. Okay. So far ada soalan?
Madam,
Ya.
kan kalau sugar phosphate backbone, kan dia negative partial charge kan? Dia kenapa negatif?
Kalau sugar
Sugar phosphate backbone,
dia ada partial charge. Mana dapat partial charge tu?
dia tak negatively partial charge.
Partial tu dapat mana-mana? Ada ke kat dalam nota partial charge?
Dah saya tercakap negative charge tu.
Negative charge je kan? Tak ada partial kan?
Ya.
So yang tadilah yang macam saya terangkan tu, mana yang cakap? Slide mana? Saya dah tak ingat dari mana. Backbone. Ni. Okay. So the backbone. Sekejap saya present sekejap. Sekejap. Okay. So the backbone is negatively charged due to negative charge on each phosphate. Okay. So this is the backbone of DNA. Dia ada apa? Repeating unit of phosphate sugar. Repeating unit of phosphate sugar. Phosphate sugar, phosphate sugar all the way through. Okay. So why is it that the backbone is negatively charged? Due to the presence of phosphate. So phosphate, it has negative charge. Okay, negative charge. Okay, negative charge, why? Because originally dia adalah OH. Bila dia dalam punya sel, dia akan ionize jadi O negative and release hydrogen ion. So that's why we call DNA as deoxyribonucleic acid. Okay, due to this property of phosphate ni. Dia akan ionize jadi O negative, release hydrogen ion. Macam tu. So that's why the backbone is negatively charged. So kalau tengok kat sini, the backbone is negatively charged, backbone, sebab phosphate bersifat dia ada negative charge. Negative charge. Due to the phosphate that ionize in the cell, in the nucleus. Okay? Nucleus within cell. Boleh?
Boleh, thank you madam.
Okay. Lagi ada soalan? Yang lain faham? Ada soalan boleh tanya? Kalau tak ada soalan saya isi mau faham Kalau tiba-tiba saya nak bagi quiz, saya boleh jawablah soalan tu 100% betul. Boleh? Saya bagi quiz boleh? Trans fat boleh contribute kepada kalau ikut kefahaman saya lah, boleh baca lagi lah kat source lain kan. So kalau trans fat, kalau asalnya cis fat dia bentuk band. Kan cis fat bentuk band. So bila bentuk band, it prevents the fat molecule from closely packed together. Kan? Dia ada cis double bond tu. Tapi kalau trans fat daripada band jadi straight. Sama juga kalau saturated fats tu kan, dia akan menyebabkan fats tu closely packed together and solidify. Tapi soalan awak ni, madam macam mana structure trans fat boleh contribute more to more than saturated fats? Jadi kena, ini saya tak sure, tapi disebabkan oleh dia boleh closely packed together itulah yang boleh menyebabkan dia form plug tu. Okay. Ada soalan lain? Okay. Any more questions? Kalau nak tanya soalan lain yang daripada subtopik lain pun boleh.
Madam,
Ya.
can you repeat the types of RNA yang messenger transfer dengan satu lagi tu saya tak dapat yang rRNA.
rRNA ni. rRNA stands for ribosomal RNA. Okay. rRNA stands for ribosomal RNA. So why is it called as ribosomal RNA? Because it is the RNA that will compose the large and the small subunit of ribosome. Sebab tu kita panggil dia sebagai ribosomal RNA. Dia adalah komponen dalam ribosome. Okay. So the komponen dalam ribosome ni dia ada large, dia ada small. So dia adalah combination between rRNA and also protein. Okay, so this component of ribosome, they are actually made inside the nucleus. Okay, they are made inside the nucleus. Bila dah terhasilnya large and small ni, baru dia akan keluar, migrate, into the cytoplasm and assembled to form ribosome. Okay, so this is ribosome. Ribosome adalah terdiri daripada protein dan juga rRNA, yang mana rRNA ni terhasil dalam nukleus. Okay. RNA adalah transfer RNA. Again, transfer RNA dihasilkan dalam nukleus. Tapi sebab dia terlibat dalam protein sintesis, so dia akan keluar daripada nukleus ke dalam cytoplasm. Kenapa nama dia transfer? Dia akan transfer amino acid yang ada dalam cytoplasm ke ribosome dalam proses penghasilan protein, dalam proses protein sintesis tu. So tRNA akan bring specific amino acids according to the code on mRNA during protein synthesis that occurs in the ribosome. Okay. So kalau mRNA, mRNA, messenger M. Dan kenapa kita namakan dia sebagai mRNA? Sebab dia message tu dikopikan. Okay, message tu daripada DNA yang daripada molekul yang besar dikopikan ke dalam bentuk yang lebih kecil, which is the mRNA molecule. Okay, so you have tRNA, mRNA dengan rRNA. Boleh?
Boleh, Madam. Thank you very much.
Okay. Lain ada soalan? So kalau tak ada soalan, apa beza king dengan cis? Cis tu ikatan double bond between carbon, kan? Cis tu ikatan double bond between carbon. Kita ada ikatan double bond. Kita ada cis double bond. Keluar. Okay. Cis. Ini kita ada trans double bond. Okay. Apa beza dia? Beza dia adalah dekat dia punya functional group ni. Ini apa dia punya hydrocarbon chain lah, perpanjang kat sebelah sini kan, hydrocarbon chain. Okay, kalau untuk cis, functional group tu ataupun group of atoms tu akan located on the same side. Same side. Okay, kalau trans, okay, it is located on different side. So sini ada X. Sini ada X, opposite side. So, itu beza cis dengan trans. Ya, kena tengok balik. Kalau king tu bila adanya double bond dekat fatty acid chain tu kan. Bila ada double bond dekat fatty acid, yang mana ada double bond, it causes the fatty acid chain tu to be bent. Band structure. So the band structure of the fatty acid kita namakan dia sebagai king. King ni disebabkan fatty acid tu band disebabkan ada cis double bond. Tapi bila awak heatkan dia, bila awak heatkan minyak ataupun ada gua hydrogenation, daripada cis dia akan jadi trans. So daripada band dia akan jadi straight balik semula. Okay, ini adalah trans fat. Okay, which is not good. Boleh Syahidah? Okay. Ada soalan lain? So kalau dah tak ada soalan, kita stop sampai sini. Okay, so saya ada attendance untuk awak fill up. Tolong isi, lepas dah isi awak bolehlah leave the meeting. Terima kasih. Sebelum awak leave, make sure untuk next week punya lecture awak tengok video yang untuk cell chapter 2 yang dah upload dah dekat YouTube. And then nota dah upload juga, nota chapter 2 dah upload dekat dalam U-Future. Latihan untuk chapter 2 akan di-upload dalam hari ni lah. So make sure you download also the exercise for chapter 2. Okay, so have an attempt to answer the question as well. So itu je lah pesanan penaja. Kalau tak ada soalan, awak boleh sign attendance dan boleh leave the meeting. Kita habis awal hari ni 1/2 jam awal. So dia akan less consume your data.
Okay, thank you madam.
Okay, sama.
Thank you madam. Assalamualaikum.
Salam.
Thank you, madam.
Okay.
Thank you, madam.
Okay.
Thank you, madam.
Okay.
Thank you, madam.
Okay, sama. Okay.
Thank you, madam.
Okay.